The use of mice for development of COVID vaccine. Celebrate the mouse for COVID research. On March 13th, we heard the then exciting news of our schools closing, which was considered temporary. However, what followed were devastating years of losses and a medical disaster that strained healthcare systems. It impacted everyone around the world permanently, from the closure of several businesses to a combined 7 million deaths. COVID has always existed in forms of mild respiratory disease but has emerged in 2019 as severe acute respiratory syndrome coronavirus 2, also known as SARS-CoV-2. SARS-CoV-2 attaches to a specific cell receptor called ACE2 found on each cell of respiratory lining, gaining access to the alveoli and multiplying. The body's response to this foreign material, inflammation, leads to damaged air sacs, where the gas exchange between blood vessels and lungs occur. This results in low oxygen concentration in blood vessels, causing shortness of breath and pain in the chest. After the outbreak on March 1st, FDA granted an emergency use authorization for remdesivir, in part because of demonstrated therapeutic efficacy against SARS-CoV infection in a mouse model. However, it was only used in severe cases to lessen the symptoms and was not seen as a long-term solution. So, the attention turned to mRNA vaccines. On August 5th of 2020, SARS-CoV-2 mRNA vaccine design enabled by prototype pathogen preparedness was published to Nature. Corbett and her colleagues working with genetically modified mice played a crucial role in the development of the Moderna vaccine. Though mice initially were not susceptible to COVID due to the absence of A2 receptors, they were genetically modified to express it for clinical trials. The vaccine works by utilizing messenger RNA that encodes instructions for cells to temporarily produce the virus's spike protein, which is recognized as foreign by the immune system. The researchers saw results eliciting strong neutralizing antibody responses and activated T-cell responses in mice, both of which prevents infection and reduces disease severity. Also included in this response were the activation of memory B cells and cytotoxic T cells, which can later recognize the virus quicker and respond accordingly. This graph from the study shows decreased PFU, unit of measurement quantifying the number of viruses that can form plaques in a given volume, as the mRNA dose increases. This research provided important evidence that the vaccine could stimulate immune defenses against SARS-CoV-2. The mRNA vaccine platform, described as plug-and-play, allows for rapid modification of genetic code to target new variants or different pathogens entirely. Additionally, trials in mice helped researchers look for potential side effects, evaluate long-term immune responses, and get accurate dosage before moving on to human clinical trials. Overall, the ability to test vaccines in animal models was a vital step in ensuring the safety and efficacy of mRNA-based vaccines, contributing to one of the fastest vaccine development efforts in medical history. My mom suffered from COVID after we were vaccinated. She was tested positive, sick in bed for a couple of days, and exhibited symptoms of COVID such as high fever, coughs, and shortness of breath. However, likely due to the pre-exposure that her body got, she healed within a week and was asymptomatic near the end. Without the early studies on vaccines with the help of mice, it would have been harder for my mom and countless others to pull through from COVID unharmed, and for that, I celebrate the mice for protecting my family and the world. Thank you.